So we're going to just dive right into chapter 13. And mostly what we'll be focusing on is viruses. So a whole chapter on viruses. How is it possible that there's that much to say about them? And we will discuss very briefly these things called viroids and prions. Because viroids is a very similar word to viruses, a lot of people get them confused, so I do need you to understand the differences. But we're not going to spend a lot of time on them because there's a lot to say about viruses. So some things I want you to think about as we go through this section. How big is a virus? Who remembers this? It is in the nanometer range, okay, so that it can range quite a lot, from like 1 nanometer up to 1,000 nanometers is a really huge virus. Most of them are much smaller. Now, who remembers how big the pores in a typical membrane filter are? Also in the nanometer range. Think about bigger than a nanometer. How big is a bacteria? So bacteria are in the micron range, right? Depending on the bacterium, somewhere in like, 2 to 10 microns, maybe smaller. So the membrane filters usually hold back bacteria, right? So the pores are going to be smaller than a micron. How much smaller? Want to take any guesses?
1,000 times.
Not 1,000 times. That would hold a lot of things. So about 200 nanometers is the pore size of a filter. So most viruses pass through that. That means that they're smaller than 200 nanometers. Most of them are considerably smaller than 200 nanometers, but there is the occasional virus that's actually pretty big and would be retained on a filter. And so as I go through this, I just don't want you to be overly concerned about that. For the most part, as a generalization, viruses are filterable. That means they pass through the pores in a typical membrane filter. What does a virus look like? What is it made of? How does it get into cells? Does anyone already know how a virus gets into a cell? If you've ever done any outreach at an AIDS clinic, you might know this for that particular virus that causes AIDS, which is what? What's the virus that's responsible for causing the AIDS syndrome? HIV. So anybody know how it gets into cells, or what kind of cell it affects? It affects T helper cells, and it specifically targets a protein on the surface of those cells, and it acts like a lock and a key mechanism, and basically the T helper cell is tricked into bringing the virus inside. This is very typical. Somehow or other, viruses have little attachment devices. They hook onto cells and trick the cell into letting them in. Can you tell viral relatedness based on symptoms? What kind of diseases have similar symptoms that you know about? Fever is a common thing. You might get fever. Would you get fever from a bacterial infection? You could, right? You can get fever from a viral infection. You might not, depending on what exactly is. So would you say that if you have similar symptoms, you necessarily have a very similar virus? Not necessarily. A lot of the things that happen to you when you're infected with a virus are general things that your body is doing to try to fight the infection. And so a lot of viruses will cause similar symptoms. Now, to answer the question of how big, I just want to show you this picture. This is a poor, well, anyone want to take a guess, first of all, what cell this is that we're looking at? It is a bacterial cell. And what is all pseudo-colored? This is an electron micrograph, pseudo-colored in blue. What are all of those little particles? And specifically, when we are talking about viruses that infect bacteria, what is the name of those special viruses? Bacteriophages. So here's our bacterial cell, and we know this is sort of a typical bacterium that's maybe 5 microns long and maybe like a half a micron wide. And so these viruses are much smaller. Here are some general characteristics of viruses. The first term you'll hear a lot, obligatory intracellular parasite. What does that mean? What does it mean if you are an obligatory intracellular parasite? You have to live, you have to replicate inside of a host cell. So when you're outside of the host cell, you're either destroyed or you're inert. Now, the thing about viruses that distinguishes them from all other forms of microbial life that we've talked about is that they have no nucleic acid. Do viruses have any nucleic acid? Do they have genes? Do they have a genome? Yes, they do. What's different about their genome? What's different about their collection of nucleic acids? They only have one kind. It could be DNA. It could be RNA. Regardless of what it is, that is their genome. That is encoding their genes. So unlike us, or even the humble bacterium, right, all of these forms of cellular life have their genome encoded in double-stranded DNA. Whether they have a simple circular single chromosome or whether they have 46 chromosomes, whatever it is. However, viruses might have DNA, they might have RNA. They do not have both. Okay? Now this nucleic acid, is it just floating around naked with no protection? What's the clothing of the virus? It is protein. It has a protein coat, okay? It's surrounded by protein that protects the nucleic acid. Sometimes a virus is enclosed by an envelope. And what is the nature of that envelope? It's not a cell wall, but you're close. What else is surrounding a bacterium? The plasma membrane. So the envelope is basically a lipid bilayer with proteins in it, much like any plasma membrane around any cell. This is not the case with all viruses. Some have envelopes, some don't. Some have spikes. Spikes generally are for attachment. Kurt.
Are some enclosed by the envelope outside or inside the protein coat?
It's always on the outside, if it's there. So we have in the center, the core, we have the nucleic acid, protein coat, and then we might have an envelope. If we have spikes, those spikes might be embedded in the protein coat if it's a non-enveloped virus, or they might be embedded in the envelope. Now, most viruses, and I mentioned this already, have a very specific host range. In fact, most of the viruses infect only one kind of cell type in one species. This is not the case for every virus. There are some viruses that infect multiple cell types, but usually it's still a pretty small number. And there are some viruses that infect more than one species, but again, it's a pretty small number and it's rare. So what does that mean in practice? If you have a cold virus, what cells in your body are affected by cold viruses? Yeah, upper respiratory cells. So if you get cold virus on your hands, do your skin cells get sick? They do not. If your cat has a cold, do you also start to sneeze and cough? Not usually. Now, it's not to say that there are no viruses that can cross the species barrier. It's just very unusual. And partly this is because the method by which the virus gets into your cell is really specific, and it relies on a very small number of keys in locks. So you have to have just the right key, and most cells have a variety of things on their surface that the virus can attach to, but it's only going to fit in certain ones. And most cells have very specific things on their surface that other cells don't have. Most species have things on their surface of their cells that are unique to that species. So looking at the question of size again, here's just a matter of scale. So over here we have our human red blood cell, which is a typical human cell can be anywhere from 10 to 100 microns. Usually they're around the order of 30 microns. We've got our E. coli bacterium, 1 micron wide and 3 microns long. And then we've got a variety of viruses, and so you can see some of them are really tiny, right? 30 nanometers. Tobacco mosaic virus, which is skinny, it's only 18 nanometers across. And then we have also, bacteriophages, typically about 200 nanometers tall and maybe 50 nanometers wide. And then we've got our good friend Ebola, this giant honking virus that looks nasty and does bad things to you. Most viruses are considerably smaller than that, though. So again, if we use this membrane filtration technique to sterilize some solution that we can't heat, then we're going to remove anything that's bigger than 220 nanometers, or 0.22 microns. So that's going to be the vast majority of bacteria, although there are some teeny tiny bacteria. And it's going to allow most viruses to go through. Now we could also try to sterilize this, whatever it is, by autoclaving, by using glutaraldehyde, by dry heat. Would any of these techniques kill viruses? I'm just going to let that question rest in your minds because I want you to be curious and go back to chapter 7 and see, can you kill viruses with these? Now, one thing I want to mention, which of these for sure do you feel confident is a method by which you can sterilize something? Autoclaving for sure, we talked about that, right? And when we say sterilizing, are we also talking about the destruction of viruses? We are. So autoclaving for sure will kill viruses. You may remember that glutaraldehyde was one of the compounds in the chemical section that we talked about as being able to sterilize things. So what do you think? Is it going to be able to kill viruses? It will. Now dry heat, what do you think? Yes or no? Kurt is saying no. Some people are saying yes. It depends how hot and how long, right? Dry heat is capable of sterilizing things. It just takes much longer than autoclaving. Okay, so let's look at what a typical virus looks like. Here's just one example. Okay, you can see that we've peeled back the protein coat for you to look inside, and all of that loose purple stuff is the nucleic acid. It might be DNA, it might be RNA, it's never both. Then we have this protein coat. It's called the capsid. That's the name of the protein coat, and it's made of capsomeres. So typically, now the viral genome is going to be small, right? It only encodes a certain number of genes, which means it only has a few proteins to play with. So in order to make this whole coat, it just makes little repeating subunits. A couple of proteins in this little unit, and then it sticks a whole bunch of them together. So all of these capsomeres are made of the same proteins, and it's just assembled into this lovely geometric structure. Now, some of the time, there will also be proteins sticking out, and they're spikes for attachment. We also might have an envelope around this, and again, the envelope is made of what? Is it protein, sugar, lipid? It is a double phospholipid bilayer. So, again, viruses might be naked. That is, they only have this genome plus a capsid, protein capsid coat. The whole thing is called a nucleocapsid. The nucleic acid plus the capsid is a nucleocapsid. If it's enveloped, then around that whole thing we have a lipid bilayer. Typically you'll see that in animal viruses, not all animal viruses, but some. And we may also have these attachment proteins that are sticking out either from the capsid itself, if it's a naked virus, or from the envelope, if it's enveloped. So what kind of shapes can we have? Hopefully from that original diagram where we're looking at size, you appreciated that there are lots of different shapes. There's sort of 4 typical ones. A very common one is this, what we call isometric or polyhedral. So every face is a triangle, a flat triangle. And there are however many faces. Very typically there are 20 faces that make this geometric structure, and that's called icosahedral, but it's not the only possible way to build a virus. It is a very easy way, though, and like I said, viruses don't have a lot of proteins to play with, so they do the easy thing. You could also have another easy way to make a coat out of very few proteins using repeating subunits is helical. So in this case, the nucleic acid is kind of in a line, and these capsids are arranged in a helical circle so that it looks like a rod at the end of the story, but it's built from little pieces going around the circle and building kind of a tower or a cylinder. There are some viruses that don't really fit into either of these, just like there are some bacteria. We know bacteria are typically in 2 different kinds of shapes, which are what? Cocci, bacillus. But we know that there are some that are all crazy shapes. They might be lumpy. We don't want to say lumpy because that doesn't sound very scientific, so we call those pleomorphic. Same with viruses. An irregular shaped virus is a pleomorphic virus. And then we've got these complex viruses where a couple of different kinds of recognizable shapes are stuck together like a Frankenstein. So typically bacteriophages have this kind of structure. Their head, where the nucleic acid is icosahedral. Then they have this helical sheath, which looks like a helical virus, and that is attached to this little landing device with spikes on it that sits down on the bacteriophage, and at this level looks like a lunar landing module. and how it functions is a lot like a hypodermic syringe. So, and we'll go over that, but unlike most viruses, the bacteriophage doesn't get inside the cell. It actually sits down on the cell and injects the DNA and leaves the protein coat outside, which makes a certain amount of sense when you think about it, because it has to go through the cell wall. It's not just going through the plasma membrane. So it's easier in a way, if you just want to think about it like punching through that cell wall, you're just injecting your DNA through all of those layers. So again, just looking at this polyhedral virus is one possible shape. We have our nucleic acid inside, again, DNA or RNA, never both. We have these triangular faces, and they're flat triangles that are made up of repeating capsomeres that are just a few proteins stuck together in some subunit and just lined up. We might have some spikes, we might have an envelope. If we look at a helical virus up close, we'll see, again, we've got our nucleic acid in the middle. We've got these repeating capsomeres. Now they're arranged in this cylinder to protect the nucleic acid. And Ebola is actually an example of this. That whole long rod there's a nucleic acid inside, and then this protein coat is built around it. Now, an enveloped virus, as I said, it could have a lot of different shapes. The inside part, it might be helical, it might be icosahedral, it might be pleomorphic, but whatever it is, that nucleocapsid, the nucleic acid plus the protein coat, has a protective lipid bilayer tucked around it. And typically, as I said, this is an animal virus feature, although not all animal viruses have this feature. So here is my favorite virus, the bacteriophage. And again, we see this nice geometrically shaped head with the genome inside. And I will just say, and I do want you to remember this, that all bacteriophages' genomes are double-stranded DNA. So I guess you could say in this way they're sort of similar to us. So if you are dealing with a phage, you know that you have a double-stranded DNA genome. There is this helical sheath. All of this stuff is made of protein except for the DNA. But these tail fibers made of protein is sitting on this injection machine. And it sits down, in this pseudo-colored EM, you can see it sits down on the surface of the bacterium. It attaches by these attachment tail fibers, and then it sits down and punches through and injects that DNA through the sheath into the cell. All right, so now we get to what is inside of the bacterial genome. Whether it's RNA or DNA, typically it's small, right? Compared to, say, even a bacteria, which has, say maybe 4 million base pairs. So here's our bacterium. There's its chromosome. There's our T4, our tiny T4 bacteriophage. Depending on whether the virus in question has a genome made of RNA or DNA, whether it's single-stranded or double-stranded, makes a difference. Now, I just told you that phages have double-stranded DNA, and we're going to talk about their mode of replication and their life cycle first because it's the simplest to understand. But there are animal viruses that have RNA as their genome, and exactly how that RNA is structured makes a big difference in terms of what happens. So regardless of what kind of nucleic acid you have, the genome has to code for proteins that will do a number of jobs. And the 3 important jobs are these. Obviously, you've got to code for your protein coat, right? Because those proteins are not ones that the host cell is going to make for you. I mean, it will, its machinery will, but it doesn't have genes to make viral protein coats, right? That wouldn't make any sense. So we have to have genes to make the protein coat, the capsid. We also have to have genes that encode enzymes that support the replication of the genome. So again, there are enzymes inside of host cells that could do some of the replication, but most of the time they can't do it all. So if we want to efficiently and rapidly make a whole bunch of viral genomes, we have to have special enzymes, and so some of the genes in the genome encode those enzymes. And then we've got to have a way to get in and out of the cell, so there are genes that make proteins to facilitate that. All right, so how do we classify viruses? Let's think about how we classified eukaryotic cells. When we talk about a eukaryotic species, how do we define it? How can we tell it's something different? Initially, a lot of the classification was done by morphology or environmental niche, but we have a better way, a more specific definition. What it eats is a good guess, but it's not the most important way that we determine different species. How do we determine something's a different species? You remember that picture of the 3 different songbirds that all looked alike? Mating. Can they interbreed? Okay. Now, we were not able to use that definition when we talked about bacteria because they don't all have the sexual reproduction mode, although they also sometimes do. Can we use any of those definitions that we use for eukaryotes or prokaryotes to talk about viruses? How do we decide that something's a new virus? Some of it is a little bit arbitrary, but really, because they're so simple and a lot of what's known about them has actually come about really recently and is part of the age of genomic sequencing, a lot of the time we use the genome itself as a way to classify a virus. So first of all, does it have DNA or RNA as its nucleic acid? That's a pretty big distinction. That actually changes its life cycle entirely. Is that nucleic acid single-stranded or double-stranded? Is there just one molecule of nucleic acid or is it segmented? Now you might think, like a bacteria, we have lots of chromosomes, so our DNA is chopped up into pieces, right? Bacteria, typically, how many pieces of DNA do they have? 1 circular chromosome. So you might think, a virus is simple, so it's probably only got 1 piece of nucleic acid, which is often true, but not always. There's one very common and annoying virus that we are very familiar with that has a segmented genome. Does anybody know what it is? We're going to talk about it later today. Something you've certainly had. Some disease that you've certainly encountered and experienced. And survived, by the way. Yes, in influenza. It has 7 segments. And we're going to talk about why that's important, especially for its evolution. So we can classify viruses on the basis of their genome. What kind of nucleic acid, what kind of structure is the nucleic acid? We can talk about it. We can look at its particle structure, which is actually pretty distinct. So is that nucleocapsid shaped like a geometric shape? Is it rod-like and is it long and skinny? Does it have some weird lumpy shape? Is it complex like a bacteriophage? And also we can look at whether it's naked or enveloped. Now, one thing I want to mention, again, is that genome structure matters, and I'm going to come back to this in a little while, but whether you're, because it's sort of a complicated but important concept, I kind of want to stress it like 3 times today. So whether you have single-stranded or double-stranded DNA or RNA is going to make a big difference in terms of what your first jobs are when you get into the cell. Because your most important task is make more genome, make more protein capsid, so I can get out and infect more cells. So let's suppose that you have an RNA genome, and it's a plus-stranded RNA genome. What does that mean? Does anybody have any idea what a plus-stranded RNA, what would that really mean in terms of its ability to do stuff? What is RNA usually doing in our cells? We have 3 kinds, okay? We've got tRNA, we've got rRNA, those are important in translation. And then what's the 3rd kind of RNA that we have? Messenger RNA, and what is it for? Yeah, it's what goes into the ribosome and gets read, right, and made into protein. A plus-stranded RNA means that it can act like an mRNA right off the bat. So what do you think happens when it gets into a cell? Do you think the cell can tell that that RNA is not one of its own RNAs? Not really. So it just starts translating the message, right? Bam. How fast is that? Of course, now you've got a problem because you have to make more RNA genomes. So how do you do that? Do we have any cellular enzymes that allow us to take an RNA template and make an RNA product? When we make RNA, typically we're making RNA from what template? Our DNA. Okay, so we're using a DNA-dependent RNA polymerase. RNA polymerase being the enzyme that makes RNA polymerase, dependent on DNA. So we're using DNA to make RNA. We don't have any way to take RNA and make a copy of that RNA. We don't do that. So one of the genes in the genome of these RNA viruses has got to be some enzyme that's going to do that. Okay? So we'll go through that again, but I kind of want to plant that seed in your mind. If you're a negative-stranded, single-stranded RNA virus, okay, so you come in and you're the opposite RNA sequence of what a messenger RNA would be, somehow you've got to make a copy that's plus-stranded, that's actually the message. And again, is the cellular machinery going to be able to do that for you? Do we have enzymes that can take an RNA template and make an RNA copy? We don't. So the virus actually has to have that enzyme packaged inside, ready to go. That protein has to be inside there. If you're a double-stranded RNA virus, which there are some, okay, then one of those strands is plus-stranded and can be used as a message. And then your main problem is, how do I make a double-stranded genome? If you're a DNA virus, double-stranded DNA goes in, where do you think that could be copied? What kind of enzyme makes a DNA copy of an existing DNA molecule? When does that happen in our bodies? When do we copy DNA? Do we ever copy DNA? Yeah, when? During cell division, during replication, right? And what molecule, what enzyme is doing that job? What's its name? It's something ACE. Ligase is an important component of it. What's the one that's actually reading? Polymerase, DNA polymerase, right? So we have a DNA polymerase. Now, the virus could hijack our DNA polymerase to make more copies of itself. It usually also has a gene for making a viral DNA polymerase just to make it much faster. Okay? So we're going to come back to that. I just want you to think about that, and this will be in the PDF when I post it online for you to see sort of what happens. Your genome is coming in somehow, and really one of the important things you have to do initially is make that genome into a message that can be used in the ribosome to make proteins. Okay, so we'll come back to that. Another way that you can classify viruses is by their transmission. Okay, so you'll probably have heard these terms. If you haven't, you will. You've probably heard of enteric viruses. They follow the fecal-oral route, my favorite, so disgusting, right? So something like poliovirus or norovirus is shed in the gastrointestinal tract, and wherever that fecal matter goes, if you don't wash your hands and someone's making your food and you get it in your mouth, then it goes through your GI tract and you get to pass it on to the next lucky person. So we've got enteric viruses that go the fecal-oral route. We've got respiratory viruses where their transmission is by inhaling droplets. Where do those droplets come from? Someone sneezed that then sits in the air for, a long time. So things like influenza virus and rhinovirus that cause the common cold. These viruses are sneezed out in droplets and then inhaled by the next host. Goes into the respiratory cells. Sexually transmitted viruses. So for instance, herpes, papillomavirus, HPV, and HIV are all examples of sexually transmitted viruses. And then we've got zoonotic viruses. Okay, these are viruses where the host is, or the reservoir is, an animal or an insect, and the way that you contract it is not human-to-human transmission, not because someone didn't wash their hands or who sneezed on you or wasn't willing to use latex. This is a transmission that occurs because an animal bites you, or a bat bites you, or you get bitten by a spider, or you get bitten by a mosquito. Okay? So things like rabies. Rabies comes from what kind of reservoir? Animals such as dogs, bats, raccoons, yes. terrifying raccoons all over the city of Seattle. I get stalked by them on a daily basis. Bunnies, I don't think I've ever heard of bunny transmission, but I'll look into that. Really? Have you ever heard of anyone contracting rabies from a bunny? Rodents for sure, right? Yeah, and there are some viruses that you can catch from like mice or rats if you have those in your attic, which I do. But again, a lot of these zoonotic viruses, once they're in you, you can't really pass them to someone else unless some crazy thing happens, like you donate organs to that person or you donate blood. Generally speaking, they're not human-to-human transmitted. Okay, so when we're classifying viruses, giving them names, this is the process of taxonomy. So typically a virus family ends in the suffix viridae. The genus and name ends in virus. And a species of viruses, a viral species, is defined as a group of viruses that share the same genetic information, so this gene sequence is very similar, and ecological niche, which is a fancy way of saying the poor host that gets infected. And that is often specific not just to which species of host, is it human, is it a dog, is it a bat, but which cell type in that host, right? Now, common names are used for species often. Subspecies, so there are, for instance, versions of the flu. Influenza is one kind of virus, and you can get subspecies that are designated by a number. And here are some examples, okay? So, herpes viridae, that would be the family. A genus within that family includes, one of the genuses is herpes virus genus. There are other genuses. A species of herpes virus genus is human herpes virus. And there are, as we probably know, 3 subtypes of this virus, HHV1, HHV2, and HHV3, which actually can be discerned on a blood test. Another example, the family Retroviridae, a genus within that family lentivirus, is the genus containing the species human immunodeficiency virus, or HIV, and we know that there are several different subtypes or substrains of this virus, HIV1 and HIV2, and we know that they can actually cause slightly different outcomes in individuals. So, the problem of growing viruses in order to study them. Why is it challenging? How do we grow bacteria if we want to study bacteria? What do? We give it some food, we give it a nice temperature, we figure out does it like salt or not, does it like oxygen or not, we put it on a plate or in a liquid, and it goes to town and eats the food and it's happy. Can we do that with viruses? Can we put them into a liquid or on a solid medium with some food lying around? They need to grow inside of a cell. So that means what do we have to provide them with? We have to sacrifice some cells to the god of the virus, okay? So here's an example of just that. These are bacteriophage. What this is a typical agar plate, which you're now intimately familiar with, covered in a lawn of bacteria, meaning that the cells are so close to each other that as they grew and divided, that what would have been a colony just touched the spreading colony of the guy next door, and you just get a complete uniform lawn or a sticky surface of bacteria. As those cells were being put onto this plate, they were infected with the corresponding bacteriophage that this particular bacteria is susceptible to. And as the cells grew, the ones that were infected also had a productive infection. Okay, so that cell, let's say, and you have to think about this, like if you are exposed to virus, do you always catch it? Do you catch a virus every time you're exposed to it? No. There are lots of reasons for that have to do with our immune system, but one of the reasons is that a virus, just because it can attach to a cell, isn't always able to infect that cell. So not every single one of these cells gets infected with a virus, only some of them do, and you can see where they are, right? Right here was a cell that was infected, and as it was infected, the virus took over, replicated, exploded the cell, and then infected all the neighboring cells. and it exploded those guys too, you form what we call this plaque. So that's one way of growing virus. You give it its correct host. Would you be able to do this if we're talking about a bacteriophage virus, we want to study it, and we put it on mammalian cell culture? So we take some cells from a human that will grow in a Petri dish. We throw in some bacteriophage, and what happens? Nothing happens. Yes, absolutely right. Nothing happens. Why does nothing happen? You don't yet have a concrete explanation for why, but I have told you that viruses are specific to their hosts, and that would be the wrong host. Basically, the particular key that virus has doesn't fit any of the locks on that particular kind of cell, so it can't get in. Okay, so when we're growing animal viruses, can we use Elanobacteria to grow animal viruses? No, we cannot. Same reason that we can't grow phage on human cells. Very typical way to do this is to inject the virus into an embryonated egg, and this provides all of the nutrients and enzymes and host cell machinery that the virus needs in order to live. Again, this works if you're an animal virus.
Wait, you actually take a live animal, embryonic stem?
Typically a chicken egg.
You eat it afterwards.
Yes, you eat the... We don't do that. Yeah, a chicken egg, which...
I'll feed you an infected chicken egg with the virus. You want it?
You inject it into the developing chicken egg, and the virus can grow.
I have kind of a silly question.
Yeah.
Are all viruses pathogenic?
No. Yes. It depends. Okay, so when you say pathogen, are you being sort of human-centered? Are you thinking if a virus infects and makes you sick versus a virus that infects bacteria and makes the bacteria sick, is that still a pathogen?
Yeah. So will there be any case where a virus may help the cells grow like that?
That's a good question. So I can say with confidence that there are viruses that don't do very much for a long period of time, and maybe ever. We've probably heard about this idea that there are latent viruses, that you can actually be exposed to a virus and have an infection but not get sick, but be sort of a carrier of that virus. Maybe you shed virus particles, but you never show any symptoms. So that can happen. I don't know of any, personally, the world is a big place. We have not explored every tiny microscopic inch of it, but I don't know of any cases where the virus is actually of benefit. But I'm open-minded. It's a good question. I don't know. I'll think more about that, but I don't think so. You think so? Okay. Awesome. Let's do some science together. Okay. So another way that we can do this is in cell culture, as I said. So we just take usually a serum, so some liquid, that a cell culture, that is mammalian cells or some kind of animal cells, can grow in. So normally they're inside of an animal, but you can harvest them from the animal. You can put them into some kind of serum that has all the things they normally would need in the body, and you can grow them that way, and you can infect them, that particular cell type, as long as it's susceptible to that particular virus, plant or animal virus, you can infect it with that virus and grow them. Okay, so how do we ID a virus? Any ideas based on the stuff that you already know? If you suspected someone was sick with a virus and you wanted to know what they had, what would you do? What kind of questions would you ask? What kind of tests would you do? Symptoms. Okay, so you would see what their symptoms are. That's not going to be definitive, right, because lots of things look kind of the same. Blood test. What would a blood test tell you? When you do a blood test, what is it actually showing you? Are there virus particles? You could look for virus particles. What else? When we are infected with something, we make, what do we make that's floating around in our bloodstream? Say again? Yeah, you can look at the count of white blood cells. What that tells you, for the most part, is more general answers. Like a white blood cell count will tell you is it likely that you have a viral infection or a parasitic infection, or nothing, or are you anemic, right? It tells you general answers, but it usually can't tell you specifically which virus you have. You could do sequencing, right? You could isolate the thing and you could sequence the DNA that you find in the sample, and there are a variety of ways to do that. You can also, what does our immune system do that's so special? What do we make to fight off invaders? We make antibodies. What are antibodies? Are they sugar, lipid, or protein? They're proteins, okay? And like I said in the very beginning of this course, proteins have a very specific three-dimensional shape. They're like the lock, and they're looking for the key. And the key is some feature of the virus that initially freaked out your immune system to make that antibody. So we can use that antibody, that specificity. We can see, are there antibodies in your bloodstream that are specifically targeted against some virus that we know you could have been exposed to? So a number of other ways that that might not immediately jump to mind are things we call cytopathic effects. We'll talk about that again. Serological tests, basically looking for antibodies against a particular virus in a patient's blood or in their tissues. And we can use antibodies to identify viruses in a variety of ways. We can look at their nucleic acid sequence, so we can use PCR, which you may remember is this, you have to already know something about the sequence that you're looking for. You make primers against the edges of it, and you PCR up that region, and if you're successful, you were right, and then you can sequence it and make sure that you were right. Or you can do RFLPs, which is basically that fingerprinting technique I told you about. So you want to review those things, you can look in Chapter 10. So cytopathic effects is something we haven't discussed already. And basically what that means is doing kind of what we do in this class, looking under the microscope and seeing if there's some kind of observable, visible change, okay? So if you just were blinded and ignore the rest of the text, this is the exact same cell culture. One's infected with a virus and one is not. Which one do you think is the sick culture and which one is the healthy culture? Sick. You think this one's sick? Some people think that. So, I mean, how would you know? Right? Looking at this, it's hard to say. I think it's perfectly reasonable to look at this and go, well, that looks kind of sick because I know bacteria are normally round. Human cells, you might think of are round. So that one looks really weird. The way to know this is obviously to have worked with human cells in cell culture a lot and to know what they look like normally. And it turns out a lot of human cells, when they're in a culture dish, they look like this. They look kind of flat and spread out with little fingers. And underneath those fingers, they have proteins that are gripping onto the surface that they're hanging onto. This is kind of what they do in your body too, right? Our body has a whole bunch of cells, and unlike a bacterial cell, right, which is round or rod-shaped or whatever shape it is, and it's happy on its own, and although we've dealt with some species that are clumpy, right, making those stains harder to do, for the most part, if they don't stick together, is that a big deal? No, not really. Now, if your human cells don't stick together in your body, what happens? Disaster, right? In fact, there are diseases that are actually the result of genetic errors in the proteins that hold cells together, okay? And they're really terrible. They actually allow you to survive to term and to continue to live, but it's a terrible existence. So a human cell has the ability, on its cell surface there are a lot of proteins that are looking for something to grab onto. And we know what those things are. Usually in the body they're grabbing onto each other or they're grabbing onto some protein that the cell has actually secreted outside of itself to hang onto. But since we know what those are, we put those proteins that they want to grab onto on the surface, and then we put the cell on top of it and it grabs onto it and it's very happy. If you infect it with a virus, it looks really weird. So something that happens is it's infected with a virus and you want to sort of think about it starting to forget what it's like to be a human cell. It's being sort of taken over by this virus. And so it loses its contacts and it starts to round up, and that's very unusual. So if we see that happening, then that is a very stereotypical feature of a viral infection. Okay, so all viruses are confronted with similar challenges, and so a overview that is extremely reductionist of what a virus life cycle looks like is they have to get into the host cell. They have to make more viruses. Then they have to get out again. And then what do they do once they're out? They find another host and they get back in. So they have a very simple life. We like to think of our lives as being much more complicated and interesting, but you could probably come up with a pretty simple reductionist human life scheme as well. So let's talk about actually the simplest version of this and variations on that theme. One of the earliest viral life cycles to be well characterized was a life cycle of one of the bacteriophages. And it runs by this lytic cycle that you may have heard of in other courses. So, how does the bacteriophage get its genome into the cell? First, there's an attachment phase, and as you saw, that lunar landing module has those tail fibers, and they're what's responsible for attaching to the bacteria. So the phage attaches by these tail fibers to its host. There's a penetration phase. So basically the phage makes an enzyme, lysozyme, that makes a hole in the cell wall. Then that helical sheath that's underneath the head but above the tail, that contracts. And like a needle, it just plunges the nucleic acid in the head of the nucleic capsid, just plunges it right into the bacterial cell. So once the genome's inside, what is going to happen next? You want to make stuff, right? What are the things you need to make as a virus? Protein and more DNA. So you've got to make more of your genome, and you've got to make more of those proteins that are part of that elaborate protein coat mechanism. So first you make all of those things. That's the process of biosynthesis. Then you put them all together. That's the process of maturation. And finally, somehow you get out, and if it's the lytic cycle, then the phage uses more of that enzyme, the lysozyme, to basically make holes in the bacterial cell wall and rupture the cell. And then all of those many, many viral particles spill out. And where do they go? To the neighboring bacterial cell. So here's a growth curve. You'll remember the growth curve for typical bacterial culture looks a little bit more elaborate than this, right? It has, for bacterial culture, what do we have? Leg. What's happening in the leg phase? We're making stuff in order to deal with the environment. We're not doing cell division. We're just making a lot of enzymes, whatever we need to survive in that particular condition. Then what's this part here? Log. And so what kind of growth is this? Exponential, okay? So we're exponentially growing. At some point, we start to plateau. What's happening here? What's that called? Stationary phase. Is cell division stopped here? No, but cell division and cell death are equal, right? So we don't get net growth. And then what happens next? Oh, death, sad. So a simpler version of this happens in viruses. We have this, on the y-axis we have the number of virus particles, or virions is another name for virus particle. The number that we can actually see, say it's 10. Now we expose them to some cells, and that number goes down. What's happening? They get into the cells, so the virus particles go away. They go inside, we can't see them anymore. This eclipse period, what's happening here? Replication of the genome and biosynthesis of the proteins for the capsid and assembly, right? Maturation of new viral particles. Then something happens. In the case of the lytic cycle, we make holes in the bacteria, all the virions escape, and we reach this point where we have our acute infection, and then it goes down again, either because we found more host cells to infect or we ran out of host cells and everybody died, right? I mean, not all the cells are dead, but also the viruses can't survive without a host. So here is the lytic cycle of a bacteriophage. A specific subset of phages called the T-even phages. And what we can see is that we've got this attachment phase where the spikes attach to the cell wall of the bacteria. The spikes are proteins. They're keys. They're attaching to something on the bacterial cell wall, which is the lock. And they sit there, make a hole in the cell wall, inject the DNA. So the DNA goes inside. What happens to the protein coat? Does it go inside the cell? It's left outside. Okay, and this is pretty much the only example. All the other kinds of viruses that we'll talk about get inside the cell whole. Okay, so that makes a big difference in terms of this step. Penetration of the DNA, biosynthesis of more genomes and capsids, and then explosion. All these viruses released from this dying bacterial cell. So if we're a bacterium and we're unlucky enough to get infected with a phage that uses this as its life cycle, what's going to happen to us for sure? We're going to die. We are going to die. But we might instead be infected by a bacteriophage that uses the lysogenic cycle, in which the DNA that's injected becomes a prophage and is incorporated into the bacterial chromosome and becomes basically latent for some undefined period of time. As long as that prophage DNA is in the host chromosome, are we going to be dying bacterial cells? No, we're just going to go about our normal existence as if nothing is happening. So this is basically the lysogenic cycle. So let's just ignore the left-hand side for the moment. If we're lucky, we have our phage sits down on the bacterium. Just like before, it makes a hole, injects its DNA. The DNA actually recombines with the bacterial chromosome and becomes a prophage. So it actually incorporates into the bacterial chromosome. This DNA gets in, and as the bacterial cell goes through its normal cell divisions, what happens to that phage DNA? It's just replicated as if it's part of the chromosome, and you could have one cell infected, and down the line, in 4 hours, 30,000 cells all having the same bomb waiting to go off, but nothing's happening. Okay? So at some point, some trigger this could go on for generations and generations, but some trigger will cause this prophage to hop out. And if it hops out, it could hop back in, or it could trigger the lytic phase of this lysogenic cycle. So it's possible, even from the get-go, for this DNA to come in and start an active infection. And if that's the case, what's going to happen to the bacterial cell? It's going to die. Now, I want you to think about why this strategy would even exist. And say again? All right, excellent. I'm just only half a step ahead of you, barely. So it's kind of obvious to think about why a virus would choose the left-hand side of this path, right? It's an easy way to make more viruses and get out and find more hosts. What if you're infecting a cell that's all by itself? That wouldn't be good, okay? Now, think about the advantages of being this kind of patient, sneaky virus. You get into the chromosome, nobody knows you're there, you're passed down generation to generation. Are you still being replicated? Are you still getting into more and more cells? Yeah, absolutely. So you're still actually accomplishing the same goal, except that if you were, say, in that one lone cell, and you decided, I'm not going to sacrifice this guy, I'm having a good day, then now we have a huge population of cells all with the same prophage incorporated into the chromosome. And if we're in a huge population of cells and we're in some sort of rich environment, then potentially coming out of the chromosome and initiating the lytic phase might be a good idea at that point. Okay? Now, I'm not suggesting that the virus knows necessarily, or that all viruses have this option. Some viruses only do lysis. That's all they do. So if they're unlucky enough to get into a cell, and they just can't contain themselves, and they initiate a lytic cycle, too bad, okay? That lone bacterial cell is the end of the line for that virus. Different environmental niches are going to be ones where either you pretty much never run out of food, so you never run out of host cells, so you might have a lot of lytic viruses. Other environmental niches are going to be mixed. You know, bad times come, there's only one cell lying around, and the good times come, we have lots of cells. So that kind of environment might foster a lysogenic phage to be successful. But it's not like the virus is thinking about that. It's just if it makes that choice, then it's successful, then it's going to be passed down. Does that kind of... Okay. All right, so just to reiterate what the lysogenic cycle consists of, the prophage DNA is incorporated into the host DNA. And this is actually interesting because the incorporation of prophage DNA maybe in a way answers your question, Kurt, which is there an example of a virus being of benefit? Now, to phage can potentially in a way be of benefit to bacteria. It might be the case that they confer some feature on that bacteria that didn't have before. Bad news for us usually. So for instance, many bacteria that are pathogenic versions of that species are pathogenic because they acquired some DNA via a virus that now they can make some toxin, or they can make some drug resistance protein. Okay? So for instance, remember we talked about the Shigella toxin in E. coli? That E. coli that makes you sick as opposed to all the normal E. coli that's floating around in you all the time? Do you remember what we said about where that toxin came from? What species it came from? Shigella. And how did it get it? How did it acquire that toxin, that protein that makes a toxin that makes us sick? It was horizontal gene transfer by the process of transduction, which is defined as what? That's when you get your DNA how? By a viral intermediate. So this isn't the only example. That super terrible toxin that Clostridium botulinum makes, where do you think it got its toxin? From a virus. Diphtheria got its toxin from a virus. Toxic shock syndrome. So are all Streptococcus species capable of giving you toxic shock syndrome? No. There's this one particular species that has this protein. It has a gene that makes a protein that causes toxic shock syndrome. And it acquired that because it was infected with a virus that happened to be carrying it. So transduction, there are two forms, and I misspelled this, and I apologize. This is not generalized. It's generalized and specialized. And your book goes into this. I'm actually going to skip over this distinction. If it's of interest to you, then you can look into it. I will not test you on this. But it's basically generalized transduction is any bacterial DNA is transferred from one cell to another via the viral intermediate. Specialized is this prophage, basically prophage-mediated result of the lysogenic cycle. So just to give you this sort of quick and dirty comparison of these two forms of transduction, if your virus is a virus that is using the lytic cycle, or it's a lysogenic virus but is choosing the lytic pathway, then one of the things that happens during that lysis is that the bacterial chromosome gets chopped up, and by accident, pieces of it might get incorporated into a maturing viral particle. And in that way, you could pass DNA to the next host. Does that kind of make sense? Now, the difference between those, so that's generalized. That's kind of like on accident. Like any piece of DNA in the chromosome might end up in the viral head. Versus specialized transduction, which is the result of the viral DNA goes into the chromosome, and only the pieces right next to it might get accidentally packaged into the viral head if the infection is productive. So this is a kind of a technicality. I don't care if you remember this. I won't test you on it, but it is interesting, and I wanted to kind of walk you through that really quick. Okay, so let's talk about animal viruses. And I think in a couple of slides we'll take a short break, but I do want to finish up this one little section first. So what does an animal virus have to do? Does it have to attach like a bacteriophage does? Yes, it still has to attach, okay? And it uses spikes, not tail fibers, but it's essentially the same idea. There's something about the protein on the viral particle that fits very precisely into something that's on the surface of the cell. So it attaches. Now, the process of penetration is different in animal viruses. What happens in the case of a bacteriophage infecting a bacterium? What is that penetration actually doing? What's getting in and what stays outside if we're talking about bacteriophages and the penetration phase? The DNA goes in, the viral particle, the protein coat, stays outside. In an animal virus, this is not true. The whole viral particle goes inside. And there are two methods of getting in, either endocytosis or fusion, and I'll talk about both of those. Now, once you're inside, there's this process of uncoating. Okay, uncoating. What do you think that means, uncoating? What are you taking off? Your protein coat. Okay, now this is something that's confusing for a number of reasons, not the least of which, uncoating, I'm so sorry. Not decoding, but uncoating. So, some viruses have envelopes, right? They have this lipid bilayer, and a lot of people think about that as being sort of an extra coat. And so this uncoating process, they think of it as the envelope coming off. And I'm just going to tell you, and I want you to remember this, even though you might not, but you have to anyway. That's not what this is, okay? Uncoating is not the envelope coming off. The envelope comes off in the first step if it's going to. That virus, if it has an envelope, that envelope fuses with the lipid bilayer of the host cell and comes off right away, okay? That raincoat, whatever you want to call it, that overcoat, that comes off in the very beginning of the penetration phase. So what goes into the cytoplasm of the cell is always the nucleocapsid, the DNA or RNA plus the protein coat, which is still there. And so that uncoating part, that happens in all animal viruses, and that's the protein coat coming off. The next thing that happens in a bacteriophage infection is biosynthesis. Do we still have to do this in an animal virus? Absolutely. And is it materially different? We still have to make genomes. We still have to make protein coats. All right, so maturation, again, is this going to be substantially different? No, we still have to assemble the genomes into the protein coats. Now, how we get out of the cell, is that going to be the same? Think about what is the same and what is different. One thing that's different is that animal cells sometimes have an envelope, right? So as they leave the cell, they've got to get that envelope back. Typically, those viruses will exit the cell by this process called budding, where they go up to the plasma membrane and they kind of steal some of it around themselves and they kind of sneak out, all right? So this is the sneaky way of leaving. Now, the bacteriophages, typically when they're leaving, they do a lot of damage, right? Bacteriophages leaving a bacterial cell explode the cell. And this can happen. Some animal viruses will leave that way, okay? So it's not the case that all animal viruses are sneaky and gentle, but some of them are.